everybody, and welcome back to Microbiology. I am your host, Mary Annarella, and we have been talking about viruses. We're in chapter 13, I believe, and I skipped over a slide here inadvertently. Sorry about that. But I did want to go over what's inside the viral genome, meaning what genes does a virus bring with it? So when a virus infects a bacterial cell, and this is our T4 bacteriophage, it's got a little piece of DNA in there, or RNA in the case of some viruses, but in this case it's DNA. But what genes are in here? What genes? And remember, and what proteins do these genes encode? Really, that's the question. And we're looking at what, viruses can have tons of genes. Something like HIV will have like 26 or something like that. But at minimum, what is required in the viral genome? What is required in the viral genome? Okay, well, first up, it has to have a gene to encode its own protein coat, to encode its own capsid. So it needs a gene that encodes its capsid, aka protein coat. I totally misspelled coat. Can you believe that? Yes, I actually do speak English. Okay, so it's got to have a gene that encodes its capsid. Remember, let's see. This is the capsid here, right? It can come in various shapes. So that's the capsid. If it doesn't bring instructions for how to make its capsid, the host cell's not going to have it, so that won't work. So it's got to have a gene that encodes its capsid. It's got to have a gene that encodes its own polymerase. Like I said, I think I skipped over this in the last lecture. If not, hey, it's a repeat for you. Here we go. Its own polymerase. Usually this will be like the Pol gene. It'll be labeled, in your books and the like. Or if you see this with coronavirus, you'll see Pol there. That refers to the polymerase that it encodes. A polymerase is like DNA polymerase or RNA polymerase. It's something that can make copies of its own genome. Because the host cell is most likely not going to do that. Third up, it needs a gene that encodes some substance that can get it in and out of the cell. So a gene that encodes a protein that will get, I'm just going to say it, in and out of the cell. So for example, lysozyme puts a nick into the cell membrane that lets the uncoating occur so that the viral DNA can get in there. It also is what bursts the cell in the lytic cycle once the viral particles have been produced, it can lyse the cell to release its particles. Alrighty. So let's see, we talked also about the lytic cycle. We went through that last time. But something that's not in these slides. I'm looking at this lytic cycle here and it looks like it really sucks. If you're a bacterial cell that's been infected by a virus, it's going to kill you. It's going to kill your cells, lyse your cells. And once the cell is lysed, that cell is kaput. That's it. no more cell left there, and that's a real drag. But there are some viral defenses against the dark arts, against the viral arts. So let's talk about those. And I need a different color. Something happy on this rainy day here. How about purple? I don't know. I guess that's a happy color. All right, so defense, bacterial defense against the dark or the viral arts. Bacterial cells have a defense against a viral invader. And it's not, it's akin to, the mammalian immune system being able to have defenses against viral intruders as well. And there are a few of these that are really important in bacterial cells. So first up, bacterial cells make a class of enzymes that are called restriction digest enzymes. Restriction digest enzymes. These are enzymes that will cut DNA at specific spots. Enzymes that cut DNA at, let's say, specific sequences of DNA. And they usually cut at spots that are sequences that are palindromes. Usually at palindromes. Okay, if you know what the word palindrome is, then you can probably guess what I'm talking about. A palindrome is, in literature, in English, in Western languages, I should say, not just English, there are other languages that have palindromes, but it's a word that is spelled the same backwards and forwards. For example, the name Hannah is spelled H-A-N-N-A-H, whether you read it backwards or forwards, right? H-A-N-N-A-H. That is a palindrome. The word wow is a palindrome. The word mom is a palindrome, although albeit they're very, very short ones, pretty easy ones too. DNA has palindromes as well. So for example, the sequence, let's say we have the sequence double-stranded DNA here, C-A-A-T-T-G. This is actually a palindrome because this strand over here is going to be the same as this strand over here, C-A-A-T-T-G. This is a palindrome here. And restriction digests will often cut at a palindrome like so. We're back to orange, probably just because it's on top. But for example, a restriction enzyme will cut it not like this. That doesn't happen. It doesn't do a double-stranded cut like this, but it'll cut it this way. Like this, so that the DNA will end up looking like this. GTTAAC, like that, and CAATTG, like that. And this will be like a 5' end and the 3' end, like that. And it'll cut it so that it won't be double-stranded anymore, like that. So for example, I'm going to move this out of the way. Yeah. You're going to have this little piece that looks like this. And a little piece that looks like that. So they're going to have that, like this one, we would call this a 5' overhang right here, like a 5' overhang. So you get two pieces, one with a 5' overhang here, one with a 3' overhang right there. This has turned out to, okay, and what it does is restriction digest enzymes won't cut methylated DNA. So really it's only going to be brand new DNA that they're going to be tempted to cut. So when a new piece of DNA, like this, viral DNA here, gets introduced into the cell during uncoating, it's not going to be methylated because it hasn't been hanging out in the cell very long. So restriction digest enzymes will chop up this piece of DNA, but not chop up the cell's chromosome because it's more likely that the cell's chromosome has been methylated at this point. So let's come back down here. So they don't cut methylated DNA. So they'll probably only cut the viral DNA. All right, we have in science, in laboratory science, used restriction digest enzymes as an incredible tool. I mean, not a day goes by. If you play with DNA in a laboratory, you are going to be using restriction enzymes to cut up pieces of DNA. So for example, let me get another color. Let's find another spot on the palette here. I have a green. Okay, let's say we've got this plasmid here. We have and you know what a plasmid is, and you know that they'll have genes. Let's say this one has an ampicillin resistance gene on it. We usually make a spot. We can engineer, scientists have engineered, I personally haven't, but I've used this a million times, popped in a set of sequences here called a multiple cloning site. And that multiple cloning site, or MCS, has spots for a variety of places where restriction enzymes will cut it. So what we can do is cut it with a particular restriction enzyme, get a little overhang here. I'm cutting into my multiple cloning site, okay, and I'm making a little overhang like what's up here, like that. I'll put my ampicillin resistance gene here, right there. And I can take a piece of foreign DNA, like let's say there's some gene that I'm really interested in and I need to make some extra copies of it. I can cut it with the same enzyme and get similar overhangs like that. Pop it in there and then cut it with a restriction digest enzyme. Cut with restriction enzyme, put piece of your favorite DNA in. But now to join these two together, we use an enzyme that you have heard of before called DNA ligase. So you seal it with DNA ligase, which joins those Okazaki fragments in DNA replication. So now we have, gene X, our favorite gene, right in there. We can pop this whole plasmid into a bacterial cell, and the bacterial cell will make a gazillion copies of the plasmid for us, and then we'll have a gazillion copies of our gene there that we can play around with. Restriction enzymes are also used for DNA fingerprinting. So used in lab science, and they are used in forensics, DNA fingerprinting. Yay. Really, really useful tool, and it is a set of enzymes that bacteria developed to help rid themselves of a viral invader. It's their defense against the viral arts. There is another tool that I wanted to talk with you about, and it is, I've spent the last couple of days trying to wrap my brain around this because I've never really found a great tutorial for how this works. But the second one, and we need a different color for this because it's special. Let's go with red. Here we go. So the second is called CRISPR. And you've probably heard of CRISPR. Well, if you've heard of CRISPR, you've probably heard it called a gene editing tool. It's a gene editing tool that originated in bacteria. CRISPR. It lets us go in and like delete portions of a gene. But it is a system that a lot of different species of bacteria apparently have developed to help ward off viral infections. So first, let me discuss what CRISPR even stands for. It stands for clustered, regularly interspersed, short, palindromic repeats. Okay, now you know what a palindrome is, right? It's a sequence that's the same forwards on one strand and backwards on the other. CAATTG, CAATTG up here. All right, that is a mouthful. Clustered, regularly interspersed, short palindromic repeats. Okay, so this part I get. Short palindromic repeats. Little sequences that are maybe similar to this that are repeated over and over and over again. That's fine. But they're clustered and regularly interspersed. Oops, I didn't put an ED on there. There we go. So there are these, I think it was like 1987 that a group of scientists, I want to say in Japan, noticed these palindromes that were repeated in Streptococcus pyogenes, which is actually the microbe that causes strep throat. What they found were these short, in the bacterial genome. Okay, so here's this little figure 8 thing here that I have is a bacterial chromosome. And they found these little, you know what, I gotta color code this differently. Hang on. All right, we're gonna color code this differently. So here's the bacterial chromosome right here. And they found these repeated palindromes that would be repeated, like every 10 bases or something like that. So these little palindromes there, these little CRISPRs, clustered. So here they're clustered and they're regularly interspersed, short palindromic repeats. Okay, so the CRISPRs are in red. These short repeated palindromes are in red. So in red. But what they found in between these, I don't know, we're going to use something a little brighter that stands out. They found unusual sequences in between these palindromes. And they found that these yellow sequences here corresponded to viral DNA sequences. And, they were all WTF all about that. Like, what the heck are they doing in there? We don't know what's going on. It turns out that associated with these CRISPRs, with these clustered short palindromic repeats, there are, we need a different color, there are CRISPR-associated proteins, or CRISPR-associated meaning Cas, CRISPR-associated. So that's what Cas stands for. So there is what's called a Cas9 protein. And what happens is that this Cas9 protein, I'm just going to draw it like this. I've been really trying hard to wrap my brain around how to best explain this to you. What happens is that you get this messenger RNA made from transcription of the CRISPR sequences. Okay, so I got to get these little arrows out of there so that I can do this. All right, so we've got this messenger RNA right here. Okay, and it's got all of these on there. I don't think you want me to color code all this, do you? That would be kind of nuts. What it will do is this messenger RNA will be associated with the Cas9 protein. And I'll put the little mRNA on here. And green is for the Cas9 protein. And what it does with this Cas9 protein with the CRISPR messenger RNA does is that, okay, it's got the remnants of these viral DNA sections in here. And when it encounters any sort of newly introduced viral DNA that looks similar to that, it cuts and destroys that viral DNA. So it cuts and destroys viral DNA that matches those yellow sequences. Isn't that wild? And so this Cas9 plus the messenger RNA will chop up an invading DNA from a virus back here. It'll chop this up. Cas9 will chop that you know what up. I almost said a swear. If we were doing a show called Drunk Biology, I would totally use all kinds of swear words. But we may have delicate ears here, and my dog is hanging out here and she has delicate ears, so, you know, we're keeping this PG-13 rated, at least. All right, so big whoop-de-doo, what the heck does this possibly have to do with gene editing? Well, these two women, oh God, what the heck are their names? What are their names? Did I write their names down somewhere? Emmanuelle Charpentier and is it Justina or Joanna Doudna? They are two women. Doudna is in the U.S. She's a U.S. scientist, and Charpentier is French. They are the two women who just won the Nobel Prize in Medicine this past year. Or I guess it was just, when did they announce them? Did they announce them in June or something like that? I can't remember. But they won for their discovery of how to use CRISPR as a gene editing tool. And here's kind of how it works. Again, I don't understand all the details of this, but I'm gonna try and explain it as best I know it. All right, so remember back here that in yellow, in between the red palindromes that are back, the palindromes are bacterial DNA, and the yellow are viral DNA sequences. They were like, hey, it's these viral sequences here that let Cas9 plus that messenger RNA recognize a virus and cut it out. What if we took this piece of DNA right here, like that, and we'll just remove it from the bacterial cells. So let's remove it from the bacterial cells here. There we go. So it's all by itself. And they're like, what if we engineered this thing to replace the viral DNA sequences with maybe some other gene that we want to cut out of a cell? So replace with, you know, gene sequences, with other gene sequences. So we need a new color. Let's go with gray. It's there. So they replaced the viral sequences with gray. Like that. That means when they get it together with Cas9 here, they're putting gray in there. this is getting messy here. Now, when this Cas9 mRNA complex here sees this gene sequence, it will cut out that gene. So it's a gene snipping tool. This will cut out the gray gene sequence. It's wild. So this is huge. This allows scientists to cut a gene from a certain place, and this is something that we were not able to specifically do before. You know, we could put cuts in someone, you know, in some cell's genome, but it was pretty haphazard, or it could happen all over the place. We weren't able to direct it. Now we can cut out genes in a specific spot. This is freaking amazing, and both Doudna and Charpentier, Emmanuel, and I forget her first name. Doudna, it begins with a J. I can't remember. They won the Nobel Prize. Nobel in Medicine. Yeah, it's the 2020 Nobel Prize. So, yay. And also the first two women have shared that Nobel in medicine. So this is a huge tool. So you do need to be aware. You're not going to have to explain all that sort of thing, but you do need to have an idea of what Cas9 does and what these short palindromic repeats are, and that it originated in bacteria. So far, it's allowed us to basically cure a woman of sickle cell anemia by going in and editing her gene for hemoglobin. So that's really amazing, and it has tremendous potential because now we can directly cut at specific spots, which we weren't able to do before. And that, my friends, is absolutely amazing. Phew. All right, so all that stuff is not in your PowerPoint. Okay, so let's see. So that's viruses in bacteria. Let's talk about viruses that infect humans. And let's get a different color, shall we? Let's go with aqua blue. All right. Viruses that infect humans. All right, they always have an envelope for one thing. Oh, no, I'm sorry. They almost always have an envelope. My bad. They usually have an envelope. They can be DNA or RNA as their genome. And they can infect us in different ways. So when they have an envelope, it's going to be similar to the lytic cycle, but in this case, the uncoating happens inside the cell. The entire virus with its envelope is brought in to the host cell by endocytosis. So entire virion brought into host cell via endocytosis. All right, and you still have to have attachment. And you still go through the attachment phase. And like, for example, coronavirus, we talked about this. If this is our coronavirus there, here's its envelope. Isn't that a lovely envelope? I just draw little swirly things. And it's got its little spike protein. The world knows all about the spike protein. We may not know anything else about coronavirus, but we know something about the spike protein. And here's the human cell here. I'm sorry, this is not drawn to scale. So it attaches to those ACE receptors, right? So this binds to there, but the whole thing is then brought in as an endosome. So this will fuse. It'll bind to the cell through the ACE2 receptors, and it will fuse with the cell membrane and be brought in its entirety, like that. So the uncoating, the removal of the envelope and the capsid, occurs inside the cell. So big difference there. Oh, I didn't draw a nucleus and all this other stuff. Okay, so in the case of coronavirus, let's talk about that. We need a different color to, oh no, I'll just stick with this, never mind. Okay, so coronavirus, I'm just gonna say CoV-2, SARS-CoV-2 to be specific, because there are a lot of different coronaviruses. With SARS-CoV-2, its genome is very interesting. It occurs in 2 parts, which is really fascinating. So I'm going to draw its genome in 2 parts. Okay, it is an RNA virus, and it's messenger RNA. Okay, so its little genome is messenger RNA. What's really interesting is that these are the early genes, and these are the late ones. And duh, the early genes are translated into protein first. But what happens is that the ribosome that are from the host cell, because viruses don't have ribosomes, they go along and they make one long piece of messenger RNA, even though there are lots of different genes in here, right? What's that called when things are, when you have one long messenger RNA? It's called polycistronic. So it's messenger RNA, it's polycistronic, and then the ribosome translates it as one long protein. Ribosome, let's say, builds it. I am doing it again. Crap, I can't believe I did this, and I'm just realizing it now. Too bad, folks. It's gonna have to be tiny. Might be, but it's been a crazy, crazy week here at Shea Microbiology. Oh, well. Okay, so the ribosome builds this as one long gigunda protein. As one megaprotein. What's really interesting is that the ribosome, and I gotta erase a little bit of stuff here. There we go. It goes along, and let's say, you know, it's right to here. Gotta get rid of some of this stuff. Let's say, you know, the ribosome is right here, building its little, building its mega protein right there. There's actually a stop codon in here. And this freaks me out. I mean, because there's a stop codon there, the ribosome stops, right? The ribosome does something really freaky. The ribosome shifts. You know how the ribosome moves 3 bases at a time? It moves 1 codon at a time, so like A, C, C, U, G, G, whatever. It does a frame shift and it moves backwards 1 base. So the ribosome moves backwards 1 nucleotide. This is freaky. This causes a ribosomal frame shift. Ribosomal frame shift, which creates a whole new reading frame. So it goes this way, and then it pauses, and it negates the stop codon. You know that when you get a frame shift, you get a whole, when you add or remove a base, you get a whole different frame shift, and then that lets the ribosome go and finish translating the whole thing off. It is freaky. Okay, so anyhow, you got this big old mega protein here. I need a different color. I can't remember what color we had here, but let's say we've got this thing here. It needs to be chopped into its individual proteins. And it's chopped into its individual proteins by one of, I want to say the last one here, I'm not sure, by one of these is called a protease. So it's cut into individual proteins by a viral protease. Sometimes they're called proteinases, but this is a protease, and it chops it up into individual functional proteins. All right. One of these proteins here is actually its polymerase. So these individual proteins, one is SARS-CoV, CoA, writing coenzyme A, CoV2's polymerase. And it is what is called an RNA dependent, so it needs RNA as its template, an RNA dependent RNA polymerase. RNA polymerases usually use DNA as a template, but RNA-dependent RNA polymerases make a piece of RNA, make a copy of RNA from RNA. So it makes a copy of its RNA genome it makes a copy of its RNA genome from RNA into RNA. In other words, it uses RNA as a template, not DNA as a template. Let's see if I can say this better. It builds RNA from an RNA template. Wild stuff. Wild stuff. So RNA-dependent RNA polymerase is rDNA pol is often how you'll see it abbreviated. Anyhow, so that's a little bit of coronavirus right there. There's something we do need to talk about before we sign off, and that is... Okay, this all looks very different here. Where is that slide? I do want to talk about the different types of viruses and how we classify them. All right, classification of viruses. You know, like how with protozoa, we classify protozoa according to the type of sexual reproduction they exhibit. We classify, that is totally wrong. We classify protozoa based on the type of motility they have. We classify fungi, my bad. We classify fungi based on the type of sexual reproduction and sexual spores that they produce. Viruses, well, they don't produce spores. We classify them first by their genome structure. So this is first. Primary way. Then within genome structure, like let's say you have a whole bunch of messenger RNA viruses, then we would base them on the shape of their capsid. You know, isometric is like this, remember? Helical gives you like a tube-like thing, that sort of thing. then whether or not they're naked or have an envelope. And I just have a little tidbit here. About 15 years ago, we had over 1,900 viral species identified. That's a lot of viruses. All right, so get rid of that. And let's just take a quick look here. Here we go. So we're looking out here. Double-stranded DNA viruses would include, herpes viruses, adenoviruses. I always said adenoviruses, but I recently heard someone say adenoviruses, so I don't know what that is. Most bacteriophage or phage are double-stranded DNA viruses, meaning that their genome is a double-stranded piece of DNA in there. Some of them are single-stranded DNA, either the plus strand or the minus strand. Parvoviruses, those tend to, there are a lot of dog parvoviruses. Some double-stranded RNA viruses, reoviruses, that is freaky. And there are two different types of messenger RNA viruses. The ones up here, which include coronavirus, which is a plus RNA strand, and retroviruses, which are also a plus RNA strand, but there's a little difference to them. Retroviruses actually get integrated into a host cell's DNA first. So that is a major difference there. But coronaviruses have messenger RNA, it's the plus strand, so it's the coding strand in them, and they actually don't need to do anything to get transcribed. They came pre-transcribed, so those genes can be translated right away. So take-home message: we categorize viruses by their type of genome. Regarding these retroviruses down here where you can't see me pointing to them, so I'll just put the slide up here. They are very interesting. No. Why am I doing that? I want to take a screenshot first, Mary. Okay, so they've got an RNA genome. But what happens is they get their RNA genome, let's say RNA is in yellow, they actually get their genome copied. So mRNA copied into DNA. Let's do DNA in black. Dun, dun, dun. All right, so I already talked about, DNA polymerase, let's think about this for a second. DNA polymerase uses DNA and makes a copy into DNA, right? Makes a copy of the DNA. RNA polymerase, most of them, take a piece of DNA and they transcribe it into RNA. So this is RNA polymerase that does that. DNA polymerase does that. And of course, an RNA-dependent RNA polymerase takes RNA and makes a copy of it in RNA. So an RNA-dependent RNA polymerase. But man, what the heck is this thing here taking RNA and going to DNA? Isn't that backwards? Isn't that transcription in reverse? And it is. Reverse transcriptase is the enzyme that uses RNA as a template and makes a copy of DNA into it. It is unique to retroviruses and one hepatitis virus, but that's a little different. It has a crappy proofreading ability, so you get a lot of mutations with retroviruses, of which, guess what? HIV is one of these. And so you get a lot of mutations with HIV, and that's one of the reasons that it's been very difficult to handle. All right, so it makes a piece of double-stranded DNA from RNA. It's unbelievable. And it becomes a provirus, meaning it integrates into the host cell's genome. And then those cells will replicate and have a copy of, for example, HIV's genome in the chromosome. Now, most of these only infect actively growing cells because think about it. If something is going to get inside a eukaryotic cell's chromosome, doesn't it have to get into the nucleus because that's where the chromosomes are. And how the heck is something going to get inside the nucleus? Except when a cell is dividing by mitosis. Isn't that when the nuclear membrane breaks up and all that? Yeah. So during mitosis, the nuclear membrane disintegrates. And that exposes the chromosomes inside to getting this provirus integrated into them. So yeah, reverse transcriptase has also been very useful in the laboratory, because being able to look at messenger RNA and make DNA from it has some advantages. For example, messenger RNA is very short-lived. It's not very stable. Let me get out of here. Messenger RNA isn't stable. It's very short-lived. It gets chopped up like crazy. It needs cooler temperatures too, and we'll talk about that in a little bit as we talk about the coronavirus vaccine that is looking promising. But it's not stable. DNA is forever. DNA is super stable. You know, that's how we could even talk about Jurassic Park as a possibility, right? Because there's still dinosaur DNA around. It's very stable. There's no dinosaur messenger RNA left over from gazillions of years ago, because messenger RNA is not stable. So if you're looking at a cell and you'd like to see, you know, let's say there are 1 million genes in here. Which ones are actively being transcribed and translated into protein? You know, they're not all constitutively expressed. So what we can do is isolate the RNA from a cell, and let's put this in a little tube, put a little cap on it right there, but, you know, and have it in aqueous form, but it's very unstable. We use reverse transcriptase to make copies of this into DNA. So that the DNA copies are DNA, and the DNA is stable. So this is all thanks to that enzyme called reverse transcriptase. And HIV has reverse transcriptase. Now, there's also something else that's interesting about the human immunodeficiency virus. You all know that HIV stands for human immunodeficiency virus, right? HIV. It causes AIDS. It's not, you know, sometimes people call it the AIDS virus. It's HIV. Human immunodeficiency virus causes AIDS. Acquired immunodeficiency syndrome. Early treatment for an HIV infection involved AZT, which was a base analog that the virus would incorporate into its genome and it would slow it down. Well, guess what? HIV mutates too rapidly for that, so that didn't work anymore. But there is now a class of drugs that allow those who are infected with HIV to live a normal lifespan, live a normal life, and get their viral numbers down to basically zero. I mean, it's why Magic Johnson is still around. Magic Johnson was diagnosed with an HIV infection and even showing signs of some immunodeficiency. I want to say it was back in the late 90s, and he's still around, and it's due to this. And here's why. HIV has a messenger RNA genome. It gets reverse transcribed into DNA, and its genes are, you know, all going to be on here in the DNA. And all of these genes are controlled by one promoter. So once it's inside the human genome, the blue would be, human DNA. This thing, well, it's a provirus at this point, right? Let's put that word in there. Provirus. When these genes get transcribed into messenger RNA, let's do it in pink. They get transcribed as one long messenger RNA. What's that word when there's one promoter for many genes? Polycistronic. And here's the deal as well. So it's a polycistronic messenger RNA. When the host cell's ribosomes translate this into protein, by the host ribosomes, it gets translated as one megaprotein, kind of like part of coronavirus's genome. Okay, so this is, I don't know what to call this, a megaprotein? Polyprotein? But it's one big protein. It is not functional until it is chopped up into individual proteins. And what does that chopping up? Proteases. Proteases cut into individual functional, I misspelled functional, functional proteins. So proteases will cut them right there. Low battery. Don't worry, I'm going to get through this. Let's see. Yeah, so let's cut them like that. Like that. There is a class of drugs called protease inhibitors. Class of drugs called protease inhibitors, and what does it sound like they do? They inhibit the action of the proteases. They stop the proteases from cutting that mega polyprotein into functional proteins. Protease inhibitors stop that from happening. So protease inhibitors stop the proteases. Protease inhibitors have been a game changer for the health of those who are infected with HIV. Game changer. And I'm not sure why we haven't tried protease inhibitors in coronavirus infections yet. I don't know enough about that now. Maybe the action of their proteases is not as strong or as necessary. But I'm working on that. So I'm going to close here because there's been so much information here that blows my mind, and I hope it blows your mind too, that whoa, this is just really amazing stuff. CRISPR, coronavirus it's ribosome doing a frame shift, protease inhibitors being a game changer. Really, really interesting stuff, and we will pick up next time and talk about latent and chronic infections from viral infections in humans. I hope you have a great day, and we'll talk about this in class. Take it easy.